Today we will discuss the last figure of the paper that we are discussing, where we are understanding the importance of VCAM on the pancreatic cancer cell prognosis. So far we have discussed the results of the in vitro and in vivo experiments, where we have find out that with the increase with the overexpression of this VCAM on the pancreatic cancer cells, uh, there is a more chances of this proliferation and migration of pancreatic cancer cells, and these results we have observed both in the in vitro and also in the mouse models or in vivo experiments. So after validating these VCAM results, the researchers need to check whether these observations of this VCAM is also been visible in the clinical clinical pancreatic cancer sample patients. So in order to validate the clinical relevance of this VCAM and CCL18, they analyzed the 134 pancreatic cancer patient samples, and out of this 134 pancreatic cancer patient samples, the three samples are shown in the panel A and panel B, where we can see that there is a group. They have categorized them into three different groups. One group that has a high expression of the CCL18, and another group has a low expression of CCL18. The the third category has a very low expression of CCL18. The second group has a moderate expression of CCL18, and similar categorizations are also been done on the VCAM score, where the a group has a high expression of this VCAM-1, and the another group has a moderate expression of VCAM-1, and the third group has a low expression of VCAM-1. And these expression levels are measured using immunohistane immunostaining of the pancreatic cancer tissue pancreatic cancer tissue samples from the patients. And after validating that the some of the pancreatic cancer affected patients has the high expression of VCAM-1 and CCL18, they wanted to check its relationship with the prognosis of the pancreatic cancer cells. And when they check this overall survival rate, they have find out is that patients that have low CCL18 has a high survival rate probability compared to the patients that has a moderate CCL18, and the patients that has high CCL18 has the lowest survival rate. And similar results they have observed for the pancreatic cancer patients that has a low level of VCAM-1 has a high survival rate, whereas the patients that has a moderate VCAM-1 expression levels has a low survival rate or intermediate survival rate, and the patients that has a high expression of this VCAM-1 has the least survival rate. And when they check the combined effect of this CCL18 and VCAM-1, what they have seen is that when both the CCL18 and VCAM-1 are low, they see that survival rates are higher compared to the case where moderate expression of CCL18 and VCAM-1 are there, the survival rates are lower, and the very high expression of CCL18 and VCAM-1 is there, then survival rate is even lower. So in this case, the number of samples that they have accounted for this analysis is shown in the figure, in the legend of the figure, and the variations could be due to the number of patients they have find out with these conditions that are mentioned in all the three cases. So together and one more thing that they have checked using the multivariate analysis is that the effect of CCL18 and VCAM-1 is independent. They are not dependent on each other, and both of them affect independently towards this PDAC poor prognosis. And so this from these results, the researchers wanted to point out is that if uh, if this VCAM-1 can be the effective marker in order to identify whether the patients has the high VCAM-1, then they might have this more proliferations and aggressive migration properties, and those migration properties can be uh, reduced to a greater extent by targeting the VCAM-1 or CCL18, thereby increasing their lifetime. So this is the outcome that one can infer one can infer from this paper. So however, you can go and read the whole papers so that you can also get some other understandings about the paper. And if you need to understand more about their experimental results, results and how their methods, you can also refer to that same paper. And this paper's reference link is provided in the descriptions. So that's all for this video and about this paper. So thank you for your time.